So this is our last experiment, ELISA or ELISA. It's actually an acronym that we'll go over, so each letter actually stands for part of a word. And this particular technology can be used to detect the presence of antibodies, which is what we're doing today, but it also can be used to detect the presence of antigens. It just depends on which sample you have as to how and what order you would set it up. So another name for the antibody is also referred to as indirect, where when we're testing for antigens, it's referred to as a direct ELISA. And this is terminology you would use if you worked in a lab like I did, where we actually perform these tests a lot. And I actually did indirect ELISA. I was actually looking for antibodies in the samples that they gave me for a particular antigen. Right? So I wanted to know if the armadillos had produced antibodies against leprosy. And I would test for that. We use this type of testing all the time. Right? You can buy it over the counter. They use it at the doctor's office. They use it in laboratories. They use it in veterinarian offices. You name it. So what ELISA stands for is enzyme-linked immunosorbent assay. So there's an enzyme linked to what's a common product of our immune system? Antibody, right? Which is actually what we're testing for today. Sorbent referring to there's going to be a color change that happened. Assay is a series of steps, and you have to follow them in order, otherwise you're not going to get the correct results. So a little general structure of an antibody is a characteristic Y-shaped. And then to represent that there's an enzyme linked to it, we have this little drawing of like a little Pac-Man. So that's our enzyme, right? This portion, the Y portion, is the antibody. And it's actually going to be the second antibody we use in our procedure. So it's referred to as the enzyme-linked secondary antibody. So our little stick drawings for our molecules here. So what is an antibody? It's a product of your immune system made by specialized white blood cells called plasma cells, which are derived from B lymphocytes, or just B cells for short. Each antibody that's produced is specific to a specific antigen. So what's an antigen? An antigen is a foreign substance that stimulates your immune system to mount a response. One of the most common responses is the production of antibodies. Not the only response, though. Right? Those of you guys in lecture, right, know T cells and all that fun stuff. Right? We're going to be utilizing a special antibody today. Our secondary antibody is actually an immunoglobulin antibody, or you could say an anti-antibody antibody, which is about a bit of a mouthful, right? Anti, I feel like I'm losing my mind. But immunoglobulin is another name for an antibody. So in this case, it's an antibody that binds to other antibodies, right? So the second antibody we use is going to bind to the first antibody we use, the primary. So this is a general drawing, almost like a stick drawing we would do for people, but for antibodies. Antibodies have major components to them that determine their ability to function. They are proteins, right? They're made by our cells, and they're secreted. Each protein is going to be made up of 4 smaller proteins. 2 light chains, and they're represented here by the shady ones, that are identical. And 2 heavy chains, which are the clear, non-shaded ones. These are identical. These 4 come together to form this whole structure we refer to as an antibody. Now, the important functional components of the antibody is the combination of both this heavy and light chain. This is the antigen binding site. This is where the antibody binds to the antigen. So the antigen would be, say, this letter A. That's where the binding's happening. The same is happening over here at B. And these 2 portions are the same, they're just mirror images of each other. So they would bind to the exact same antigen. And I'm trying to remember. Yeah, we're going to do antigens bind to antibodies. So at A and B, right there. So in our body, our immune system would interact with this antibody when it's bound with antigen. And there's lots of different components of our immune system that will interact. The portion of the antibody that it interacts with is down here at the C portion. And this is commonly referred to as the FC portion. So it's the fraction that actually crystallized when they were studying it. But it's also pretty consistent from antibody to antibody. This portion is going to be the same. So this is where our secondary antibody is going to bind to the first antibody. Because molecularly these are pretty much the same for all antibodies, we can use an anti-antibody that's going to bind to it. So if we were running this test, we would get a serum sample from somebody. In their serum contains lots of different antibodies, and this is denoted by the different colors. So notice each antibody has a different color because each antibody is unique. It binds to a specific antigen. So how are they different? Right? So they're different colors to represent that they are different. But when you look at the structure of the antibody, as I said, the bottom portion is pretty consistent. It's that portion up at the top that binds to the antigen that's variable, that varies from antibody to antibody. Right? Both the heavy chains and light chains are different. And that makes each unique antibody that unique, that it's specialized. In our body, we produce 5 different types of antibodies. We produce IG, stands for immunoglobulin, A, D, E, G, and M. For those of you guys in lecture class, you should know a lot about those, but we're just going to mention them here. And it's that binding of the antibody is really down to the molecular level. So when we talk about exactly where the antibody is binding, it's referred to as an antigenic determinant or an epitope. So it's very specific as to where these antibodies bind, right down to a molecular level. We're talking about 10 or so amino acids that this antibody binds to on, say, the surface of a bacteria or within a protein. Right? Something as small as a protein can be very antigenic. It can get our immune system to produce antibodies. So there's 2 different forms of liquid component as it relates to your blood. Plasma is when your blood is not allowed to clot and they spin off and get the cells out and you are just left with that liquid phase. That's referred to as plasma. As an immunologist, most of us do not like to work with plasma. The problem with plasma is those clotting proteins that would normally make the clot are in that liquid phase because the blood wasn't allowed to clot. Those clotting proteins can interfere with immunological testing. So typically, if we had the choice, we'd rather get what's called serum. And the difference between serum and plasma is that serum is a liquid phase that's taken after the blood has been allowed to clot. So all those clotting proteins are trapped up in the clot, and they won't be in the liquid phase. Your antibodies, on the other hand, are in that liquid phase of whether it's plasma or serum. They're going to be there. The problem with plasma is that those clotting proteins are also there, right? So it can interfere with these tests. So when I was in research and my boss only wanted to take 1 blood tube from the armadillos, I was like, great, plasma. But that's okay, right? I can make it work, but I'd get more accurate results if he would have took 2 tubes so that I could have a tube that was allowed to clot and I could get some serum. So when people refer to serological tests, they're usually using serum, right? That's why it's referred to as serological. And they're looking for stuff that's in that other than clotting factors, right? Because the clotting factors are going to be in the clot. But antigens will also be in that liquid phase, as well as the antibodies will be there. So which are we doing? Are we doing antibody detection or antigen detection today? We're doing antibody. So we're doing indirect, right, not direct ELISA. So Bio-Rad has a great set of animations that I have a link to on Blackboard for you guys. And I'm going to show you in a second. So they have both the animations there for you, and I highly suggest looking at comparing the two. So they have the antigen detection, which is the direct, and this is where a mixture of antigens directly from the patient's serum, followed by a pure, commercially available primary antibody is used, right? Where we know what the antibody binds to, right? We're looking for that particular antigen. So examples of that is the rapid strep test, right? So when you go to the doctor's office, they're actually testing for the presence. In this case, they're doing a throat swab, right? And they're looking for that antigen in that test. Ovulation tests and pregnancy tests are actually testing for a hormone. But that hormone could be considered an antigen, right? Your immune system could make antibodies against it. Of course it doesn't. We get these antibodies from other sources, right? We use it outside of the body in a test. So you're testing for particular hormones when you're doing ovulation and pregnancy testing. But you're using an antibody to detect the presence of that antigen, right? That hormone in this case. Drug testing is done a similar way sometimes. HIV antigens, if they're looking for the antigen, they can use antibody specific. Various flu, when they do the rapid flu test in the doctor's office, again, could be detecting for the presence of the virus. Indirect ELISA, or antibody detection, in this case, we're starting with a purified antigen. And then it's the sample from the patient that contains the antibodies that we're looking for. And so this is often used, especially when antigen levels will tend to be really low, right? Or you're less likely to detect the presence of the actual causative agent. You're more likely to actually be able to detect the presence of antibodies. So some tests that do that is they'll do HIV testing for the antibodies. Has the person made antibodies against it? When you take your dog to the vet and they draw that blood sample for heartworm testing, they're actually testing for the antibodies. The worms themselves are huge, right? Really hard to detect. Hepatitis viruses, if they're testing whether you have the antibodies or not. Now, this could be because you've been vaccinated, right? Or because you've had the infection. There's no way of knowing which is which unless you tell someone, I just got vaccinated. Which is one of the reasons when you've been vaccinated for hepatitis, you can't donate blood right away. Because they'll see those antibodies in your blood, and they might just pitch your blood thinking you might have hepatitis. But they do extensive testing on blood that's donated so they don't pass disease on to someone else. Any questions on that? And a lot of times in the hospitals, they're doing combined tests now where they test for both antigen and antibody. So they get more accurate results. All in one test. Pretty amazing stuff. So, Peter just added this slide and I didn't realize it. But as you can see, we can read through it together. HIV targets your T cells, a particular population of your T cells referred to as your CD4 helper T cells. So you can see an infection. So this is the cell count, right? And here is the viral count. So cell count decreases, of course, as the virus increases, right? That's acute infection. Over time, as you can see, the virus just gradually increases. And the person's number of T cells just continues to decrease. And the bad news is that once you reach a particular level, then your T cells are basically gone. And they're at such a low level, it's like they're non-existent, and they can't protect you. That's when someone's reached the status of what we refer to as AIDS. What AIDS stands for is acquired immunodeficiency syndrome. At one time, they didn't know what was causing it. Other things other than HIV infection can cause your immune system to be deficient, right? but HIV is one of the major players, right, in debilitating people's immune systems. So the problem with that is your immune system isn't functioning properly, you're deficient, and so you're more susceptible to opportunistic disease. So it's usually actually some other disease that your body can't defend against that you actually die of. Anyone know the most common cause, secondary infection that causes death in AIDS patients? Pneumonia. Pneumonia is the #1 killer of people with AIDS. So does he have the... So initial HIV testing uses an ELISA test for both types. There's actually 2 different types of the virus, type 1 and type 2. And so whether you're producing antibodies and whether you're producing this particular antigen. One HIV test is both sensitive and specific. Additionally, screens exhibit 0.3% false negatives and 1.5% false positives. But follow-up testing improves the specificity to approximately 1 false positive in every 250,000 assays. The very low risk population. So they do additional testing, right? Because you don't want to give someone this prognosis unless you're absolutely sure. Anti-HIV drugs target the replication of the virus's nucleic acid, help stop its attachment to the cells, its ability to copy its DNA. It's actually RNA, isn't it, I think? And it's a reverse transcriptase where it goes from RNA to DNA to RNA. Stopping it from integrating into the host genome and stopping it from making its proteins. So they throw lots of different drugs at it to try and stop this virus. So I wanted to show you guys that animation. Oh, I got out of my course site. So in the Lab 12 folder at the bottom, way down here, is a link to the animations. I hit the wrong one. There we go. So, see, antigen detection or antibody detection. And when you click on them, see how it has the play button? You actually have to keep hitting that play button. See how it keeps flashing? And there's no audio, right? So if you want to see it in motion, but I have pictures from each step in my PowerPoint that I'm going to go by instead of playing the animation. So I like this analogy. We're going to make an antibody sandwich, right? So we're going to start with an immobilized surface, which is going to be your plastic well. We're going to add purified antigen. Then we're going to add the primary antibody. Now, your patient sample is either going to have it or not have it. Our positive control is absolutely going to have it. Our negative control is not going to have it, right? So we're really testing for the presence of this antibody. Is it there? Is it not? So it binds to the antigen. Then we're going to add our secondary antibody. And what's special about this antibody? What does it have attached to it? What's the name of what does ELISA stand for? Enzyme linked, right? So this one has the enzyme linked to it. And that's what it's going to help us visualize if all of this is present, right? If you had antigen binding, then the antibody, the primary one we're interested in. And then the secondary one with the enzyme. When we add our colorless substrate, we're going to get a color production because of the presence of the enzyme. So it's going to be blue. It's going to be pretty. Other enzymes may produce fluorescent products, right? Fluorescence is very common nowadays, or chemo-luminescent products. We're just going to be dealing with a color. So these are some examples of where they've actually used antibodies, right, with different fluorescent tags on them to be able to stain different portions of cells. So as you can see, the nucleus in all of these is blue, right? Because the tag they used had a blue fluorescence to it. And then two other ones, right? You can see the green over here, the green and the red, right? They're binding to different components. So this is expression of lymphatic endothelial cell markers. The green labeling panels, A and B respectively, show a particular localization of a protein. Right, so they can actually label actual proteins and see where they're being expressed in the cells. Pretty amazing stuff. Right, so I have lots of pictures like this in my office too. I have a whole poster that looks like a rainbow of all the different pictures that scientists have taken using fluorescent dyes on cell samples and stuff like that. So, really cool stuff. So for today's procedure, we only get one color. No fluorescence. Not today, except for our E. coli. You're going to start with a microtiter strip, right? And I'll give you one, and when we're done with it today, it can just go in the trash. We're just dealing with proteins today. Nothing pathogenic. You don't have to wear gloves. You can if you choose to. But it's not anything that's going to hurt you. It's actually all antibodies. So we're going to start with our wells. We're going to label them, and I have an example in each one of your bins on how to label it. We're going to write right on the tube. Then we're going to add our purified antigen. This is going to be your green tube in the rack that I'm going to give you. And then we're going to incubate at room temperature for 5 minutes. So these molecules, right, are going to stick to the inside of those wells. Anything that doesn't stick, we want removed. So we're going to do a series of washes, and I'm going to show you how to properly perform those when we get ready to do that. So anything that's unbound, we're going to shake out onto stacks of paper towels. So notice some of them are missing, right? They were the ones that didn't stick. They got washed out. Then we're going to add the primary antibody. But this is what we're testing for, right? So wells will be washed a second time before we add these. So we want to make sure we wash out anything that's unbound. Why do we do the washes? We want to remove anything that's not binding, because that's going to help, that's going to create cross-reactivity where we get false positives, right? And we don't want that. Right? So remove unbound agents. What would happen if you wash too vigorously? We're going to wash out all the antigen, right? And we'll end up with false negative results, right? So we don't want to wash too much, right? What if we don't wash enough? We're going to leave stuff behind that's not bound, right? And we're going to end up with false positive results. And this creates a problem on how our assay is run, right? And that these guys are floating around and they're going to outcompete the other reagents we're adding in there, which can lead to false positives, even false negatives, and messing up the sensitivity of our test. We're not going to do, as you'll notice, the blocking agent for this exercise is not necessary. Normally what this would do is coat the other sections of the well where the antigen isn't to stop for nonspecific binding. So the next thing you're going to do is you're going to add your positive controls to your positive wells. You're going to label 3 of your wells positive, and it's going to be our purple tube. So for our positive controls, why is it essential that we do a positive control? If a positive doesn't come out positive, do we trust our results for our unknown sample? No, right? What has to be in this? What are we testing for? What's going to be in our positive control? The primary antibody, right? The primary one. The one that actually binds to the antigen is what we're testing for. why do we do positive controls? Make sure that the test is working properly. What if we get negative results in all our positive wells? Test isn't working, right? This holds true for like good old pregnancy tests, right? Now they have the little digital readouts, but if you remove that, there's 2 lines if you're pregnant. One's the positive control, and the other is your test sample. So if it's negative, you always still have 1 line, your positive control. If you don't have that 1 line, the test is going to tell you error. Redo it. And it didn't come out as expected for its positive control, so you can't trust the unknown sample, your sample. Okay? So our controls have got to come out as expected, right? Otherwise, our test isn't valid, right? Something went wrong, right? You maybe washed too much. You added the wrong reagents, right? The reagents are expired. So this can happen. So for our positive controls, it will contain the primary antibody. It will bind to the antigen as you see in this picture. For our test sample, one of them is going to be positive and one's going to be negative. You've got to find out who's getting the good or bad news, depending what you pretend you're testing for today. Right? Our negative wells, what are they going to be lacking? What are we testing for in this test? Primary antibody. So therefore they're not going to have the primary antibody, right? Not unless I messed up, and I'm pretty sure I didn't. So this is going to help us prove that only the primary antibody will bind to that antigen, because we should get negative results if there is no antibody, right? Why do we do negative controls? Make sure it's working properly, right? So we can't get positive results without that primary antibody. Because we don't want any false positives, right? We want to be sure it's only that antibody that's going to cause a positive result. So what are possible causes of positive results in all your negative controls? You didn't wash out. Right? Remember, if it's not bound, it's going to create problems. We only want stuff that's binding to be washed out. To be stay. Everything that's not. You might have contamination or cross-contamination. And so that's why it's going to be important that you wash them properly and you blot properly. So I'll show you how to do that properly when we get to that. How would positive results in all the negative controls affect your interpretation of the results given by the patient samples? It makes them invalid, right? Negative should be negative. If negative is positive, something went wrong. You don't know what went wrong, right? So you can't assume about that patient sample. You'd have to repeat the test. So, what's the only thing that should be in the wells for our negative controls? What's the only thing we put in there that binds? It's the very first thing we put in the wells. Antigen. Right? No primary antibody binding to this, right? Nothing should bind. So just the antigen. Primary antibodies absent. Now, you have 2 patient samples, patient number 3 and patient number 4, mostly so you can distinguish those numbers, right? So we're going to add those to the wells, right? We're going to let it sit for 5 minutes. We're going to actually do this in triplicate. You're going to have 3 wells for your positive, negative controls and your 2 patients. Why would you do it 3 times? Well, if you do something once, what's the chances you're going to make a mistake? pretty high. If you do something 3 times, less likely, right? So gives us more accuracy, more precision, and we could even calculate standard deviation if we needed to. These color changes can actually be measured on a spectrophotometer. You can actually get a number for the amount of color. We're just going to say positive or negative today. But in the testing that I would run, I would actually run out a standard control curve where my control, positive control, is diluted to a certain amount, right? And I knew that concentration, so then I could use that with a spectrophotometer or plate reader. You put the whole little plate on, and it would give me a number readout for each one of the color changes. And I could calculate. So what might explain inconsistent results? Say in the patient samples, one well is positive, next well is positive, the next one's negative. You probably goofed in pipetting, right? Maybe you skipped a well, right? Poor technique, or it could be poor sensitivity of the test. And you want reproducible, right? So the validity is cast some doubt, right? If somehow you end up with some positive and some negative, and not all uniform. So, qualitative assay. So we're just going to say positive or negative, right? They either have the antibody or they don't. So, patient who's test positive have the antigen, which we added, and the antibody, that primary antibody, is binding to that antigen. If they're negative, what's going to be in those wells after we wash? Just the antigen, right? Because negative ones won't contain the antibody, nothing to bind to the antigen, right? Anything that isn't binding will get washed out. Right? So if we had high-powered molecular microscopes, we could see this. But you're not going to see this, right? You're just going to see liquids going into these tubes. So here's an example of a microtiter plate, right? 96 wells. And you can see the variation in color, right? So a spectrophotometer would actually be able to tell the different levels. You can see that this is the negative control. See how there's no color? Here's the positive control, but it's been diluted, right? So very low concentrated to high, to a level that the scientist knows, right? And can use that to figure out how much is in each one of these wells, or what we would refer to as a titer when you're talking about antibodies. It's a concentration. So, as I said, we're going to wash, right? We're going to wash twice. Right? What happens if we wash too vigorously? We're going to wash out the antibodies, right? Maybe even the antigens. We'll end up with false results. If we don't wash enough, though, right, stuff is going to get left behind, can interfere with the next steps. So the next step, once we've got antigen and possibly primary antibody present, we're going to add the secondary antibody. And this is the important one. So remember, negative samples, just antigen. So here's our secondary or conjugated, because they've added that enzyme to it. It's going to be your pink tube that you get. And so notice the little Pac-Man antibodies. What are they actually binding to? And this is probably the best one to see right here. They're not binding to the antigen, are they? This is the antigen. There's the primary antibody. There's a secondary antibody. They're binding to the primary antibody. Right? This is helping us detect the presence of that primary antibody, because the secondary antibody only binds to antibodies. It does not bind to the antigen. Make sense? So this is that sandwich effect. Antigen, antibody, antibody. So what's it binding to? the primary antibody. So what would a negative sample look like? After we wash, it's just going to be antigen. Because there's no primary antibodies here, nothing for the secondary to stick to, right? So it's going to get washed out. The last step, which you'll have to ask me for, I keep it in the fridge to keep it out of exposure to light. This time, because you could potentially have a lot of stuff in here, you're going to wash 3 times. All right, again, we don't want to wash too much. We'll remove stuff, end up with false negative results. If we don't wash enough, right, we can leave stuff behind and create false positive. So, how many washes before you get your substrate? 3. And I'm going to ask you, did you do your 3 washes before I give you your substrate? The substrate is going to be in an orange tube. We're going to add it to your solution. If the antigen, primary antibody, and secondary antibody with that conjugated enzyme is present, the enzyme will interact with the substrate. There should be some foil in your racks. This is light sensitive, so just cover it up after you add your substrate for about a minute and then take a peek. And you'll get a blue color if it's positive, if all these things are present. No longer than 5 minutes. Literally within a minute, we usually see the color change. So, why is it important not to mix antigen antibody or antigen antibody solutions during the experiment? You're going to mess it all up, right? There's a reason why these are separated from each other. And this is also why it's important that we don't overflow our wells, right? And so we don't get that mixing between the wells. We don't get that cross-contamination. Most likely ending up with false positive results than negatives. We want these reactions to be happening separately. So, without washes, right, these things would stick around that aren't binding, and they would compete with the other stuff that we're adding. So, and it also would create these reactions in the tubes ahead of time. We want it only happening on our immobilized surface. So dot blots and western blots are similar to ELISA, and that the sample, in this case, is embedded onto a membrane. And then that's the surface, and then the antibodies are added to help you visualize. But in this case, a lot of times, why they're on this strip is they've been spread out, maybe during electrophoresis, to separate the individual proteins from each other, so they can tell what exactly, which protein, what size the antibodies are actually sticking to. So this is used a lot in research. It's much more specific testing and used a lot for protein assays more than anything else. So affinity chromatography is kind of like indirect ELISA, and that you're dealing with a purified target. It's immobilized on beads, and then we use a specific protein to pull the particular ones off of the binding, and allows them to separate out substances from one another. You didn't put any pictures of those in there. So a lot of testing in your doctor's offices. When you go to the drugstore, when you go to the veterinarian, right? When scientists are doing particular, being able to look at particular proteins and antibodies, they're using this wonderful ability of antibodies to bind to specific molecules, right down to that molecular level. So our immune system produces these wonderful molecules. So do most animals. And to give you an example, in today's experiment, we're going to be using a chicken antibody to then bind to an anti-chicken antibody produced in a rabbit, and then an anti-rabbit produced in a goat, right? So when you take antibodies from us and you put it into another animal, they see it as a foreign protein, and they'll make an antibody against it. So same is true for the chicken and the goat and the rabbit. You put antibodies from a goat into a rabbit, the rabbit's going to make antibodies against that goat, right? And so that's how they made the antibodies for our testing that we're going to do today. But you guys can pretend you're testing for whatever you want to pretend. Just got to be an antibody, right? So does the person have HIV antibodies? Do they have hepatitis antibodies? Does your dog have heartworm, right? These are the type of questions you could potentially ask. And what I want to know today is which number came out positive. Test sample 3 or 4.